To reach a developing insect larva hidden inside a plant gall, a parasitic wasp has to bypass a fortress. The wasp must navigate specialized chemical defenses, bore through tough plant fibers, and calculate the exact depth of the internal chamber to lay its egg. Perfecting these tools requires a strict biological compromise known as multifarious selection. Evolving the specific chemical sensors and precise ovipositor length to penetrate one type of gall inherently degrades the wasp's ability to successfully attack galls with different chemical signatures or structural depths. These intense environmental trade-offs create a rigid rule in parasite evolution. Survival heavily favors narrow host specialization. Adapting to two different environments at the same time demands too much biological energy. Therefore, a true generalist parasitoid, one capable of navigating dozens of distinctly different gall environments, should theoretically not exist. Yet, the historical record contains a glaring anomaly, Ormyrus labatus. This solitary idiobiont ectoparasitoid has been officially documented attacking over 65 distinctly different types of oak galls. For decades, 20th century taxonomy relied almost entirely on morphological keys. Because these wasps shared identical physical characteristics, from wing venation to body shape, researchers grouped them into a single, supremely adoptable taxon. The red dots denote O. labatus morphology. The blue dots mark other Ormyrus species. This map displays the collection regions for these physically identical specimens. The red dots represent wasps matching the O. labatus description, spanning widely varied climates and habitats across the United States. Modern data shatters this classification. The evidence identifies Ormyrus labatus as a taxonomic illusion, a complex of 16 to 18 highly specialized cryptic lineages. To solve this biological mystery, we have to deconstruct the modern data matrix and see how scientists detect invisible speciation. The first layer of the matrix is pure genetics. Researchers extracted and sequenced the mitochondrial COI gene from the collected wasp specimens to establish a baseline of relatedness. The sequence data was fed into ASAP, an algorithm scanning for mathematical gaps where genetic divergence exceeds normal species variation. To verify, researchers ran the parallel BPTP algorithm, analyzing phylogenetic branch lengths to calculate the probability of true speciation. To avoid overcounting, the researchers applied a strict conservative methodology. In the rare cases where the ASAP and BPTP models disagreed, they defaulted to the model predicting fewer species. This phylogenetic tree maps the genetic divergence. Sequences diverged by 6.5 to 15.6%, guaranteeing prolonged reproductive isolation in insects. The analysis definitively slices the O. labatus morphotype into clades 9 through 36, establishing them as distinct groups. DNA proves these lineages are no longer breeding, but molecular data alone cannot explain the ecological pressures keeping them apart in the wild. By adding the ecological field data to this tree, specifically the gall host and description columns, the actual mechanisms of isolation become visible. The researchers found distinct genetically isolated clades living and operating sympatrically, meaning they exist in the exact same geographic locations. Genetic isolation in these wasps is driven by microenvironments rather than physical distance. Look at clades 35 and 36. Both exclusively attack acrospis galls and are sympatric in locations like Urbana, Illinois. But the tree section column reveals they partition the niche. Clade 35 targets the quercus section, white oaks. Clade 36 strictly targets the lobata section, red oaks. Even when the target gall morphology is nearly identical, the chemical or structural differences of the host tree itself act as a hard reproductive barrier. Biological isolation exists in time as well as space. Phenology, the seasonal timing of life cycles, creates invisible temporal walls between populations. Returning to the matrix, we see clades 20 and 22 attack the exact same physical microenvironment, a mid-rib leaf-swelling gall known as C. pigra. The tree host column shows clade 20 operates on red oaks, while clade 22 targets black oaks. But this dot plot tracking adult wasp emergence reveals the actual barrier between them. Their life cycles are staggered. The adults from these two clades emerge a full month and a half apart. Because adult parasitic wasps have incredibly short lifespans, this phenological mismatch prevents adult wasps from clade 20 and clade 22 from ever encountering each other to mate. Temporal isolation locks in speciation as effectively as an ocean or a mountain range. The staggered seasonal timing permanently severs gene flow. When analyzing large datasets, the standard scientific urge is to hunt for a single unifying rule, a universal driver of speciation, like a specific gall shape or a strict adherence to one tree species. The Ormyrus data rejects a universal rule. Instead, multifarious selection forces each lineage to specialize opportunistically, based entirely on whatever local niches and combinations of traits are available. Looking at clade 25, we see strict physical specialization, targeting exclusively leaf-based galls on the swamp white oak. Contrast that with clade 32, a lineage jumping chaotically between different gall types, buds, catkins, and woody stems, across both red and white oak sections. If we look back at the emergence timeline for clade 32, the data explains how. This clade cycles through 3 distinct sequential generations per year. Each generation is perfectly timed to exploit a different seasonal gall as the year progresses. Shifting to a new host requires a lineage to adapt to completely different combinations of physical architecture, chemical defenses, and temporal emergence windows just to survive. Zooming out to view the entire integrated matrix, the historical concept of a generalist, labotus, collapses under the weight of its own complexity. Disentangling these cryptic species has immediate practical consequences. Applied entomology and biological control programs rely entirely on accurate host ranges to safely manage invasive agricultural and forest pests. If an invasive gall wasp is threatening a forest, releasing the wrong cryptic clade of an assumed generalist parasitoid guarantees a complete biocontrol failure. The wasps simply will not attack the intended target. The deconstruction of O. labotus belongs to a wider shift where molecular tools dismantle historically accepted generalists across several families, from the tephritid fly parasite Diachasma aloum to the beetle parasitoid Aesocodes lucens. Relying strictly on shared morphological traits actively blind science to massive, hidden reservoirs of specialized biodiversity. True generalist strategies in parasite evolution are exceedingly rare. The energetic and evolutionary costs of acting as a jack-of-all-trades simply do not scale against highly evolved host defenses. The myth of the generalist parasite is perpetuated by the taxonomic impediment, a lack of funding, personnel, and modern tools to properly classify the most species-rich groups on Earth. Proving a modern species hypothesis requires rigorous integrative taxonomy. It demands uniting molecular DNA sequencing with deep, localized ecological tracking, precise mapping of physical host traits, and strict observation of seasonal phenology. Whenever the historical record suggests a highly adaptable generalist parasite, it should be treated not as a settled fact, but as an invitation to hunt for the invisible boundaries of speciation.